All right. Thank you, Suba Sensei. It's a pleasure to be here. I've been in Japan now for 5 days. I still have jet lag, but it's just a fascinating place to visit. It's so much fun. So again, thank you very much. Let me just start with a bit of an introduction. I am going to talk about allostery today, but for those of you that aren't as familiar or you don't work with proteins, it might surprise you to learn that you could pick any human cell, a human embryonic kidney cell. It's microns across in size, microns, small thing. But every human cell contains on the order of 100,000 different kinds of proteins, if you count post-translational modifications. Huge diversity, 100,000. The population of proteins in a human embryonic kidney cell is on the order of 1 to 2 billion. So that's like 30 Tokyos. So think about that complexity, right? That's one cell. Now think about the interactome. Every single one of those billion proteins has an interactome, and we know this from mass spec, that is on the order of dozens to hundreds. How does it do it? How does a cell accomplish or get anywhere with that kind of complexity? And the answer is allostery dynamics, right? The cells, the proteins in these cells do not have single configurations or structures like a puzzle piece. Not if there's an interactome of dozens to hundreds, right? They have to have extraordinary capacity to be dynamic on an atomic scale in order to accomplish function. And I'll give you an example momentarily of an enzyme, and we'll talk about a reaction coordinate pathway and the dynamics that are needed for the protein to achieve the chemistry. But then there's also the dynamics that are needed for the protein to achieve interactions with different partners to accomplish the biochemistry. Okay? And so I'm going to pick up on what Adnan talked about today and really sort of motivate the problem of allostery. And I'll give you an example first about an enzyme. I'll talk about some of the complexities of allostery and how those complexities achieve function in the enzyme. But then I want to take the conversation to something more practical, and that would be the problem of drug discovery, okay, in a class of proteins called GPCRs. And then I also want to talk about, and Suba Sensei mentioned this morning, I also want to talk about nanobodies. So I'll talk about nanobodies, I'll talk about fragment-based drug discovery in GPCRs, and really the common theme throughout all of these things is allostery. So this is an example of an enzyme. This particular enzyme, Adnan mentioned it this morning, is about 300 amino acids. So for those of you that like Lego puzzles, right? One of these proteins has, if you count the water molecules, 10,000 or so atoms. So if you like Lego, the Millennium Falcon used to be one of the largest Lego sets you could buy. And it's kind of on that order of complexity, 10,000 atoms, right? And so you can see in the cartoon, this is just a sort of a backbone schematic of this dimeric enzyme. So one half is 300 amino acids, the other half is 300 amino acids. And there's some kind of three-dimensional structure that you can see from this cartoon. And then the blue dots represent water molecules. And what you'll notice is that as Adnan showed, although the backbone structure is quite similar along the reaction coordinate pathway. So as the enzyme undergoes its dynamic machinations to do catalysis, right, as it goes from one state to another, the backbone is reasonably constant, but the side chains move, the waters reorganize. And so this is allostery, living allostery. This is allostery happening in this enzyme. And so in this cartoon, you'll notice we start with an apoprotein. Apoprotein means the protein is in its ground state and it hasn't received the substrate. The substrate here is a tiny molecule. This tiny molecule is fluoroacetate. And the job of this enzyme is to cleave a carbon-fluorine bond. And for those of you that are chemists, that's kind of interesting because a carbon-fluorine bond is a very robust, high-energy bond. And remember, this machine has to do the cutting, has to do the catalysis with nothing other than thermal energy available to it. And then you'll notice that as the substrate binds to one of the two pockets in this dimer, so here you see a cartoon of the substrate binding in this pocket, the other half, the empty half, so the empty protomer, suddenly becomes more dynamic. And that's shown by these red blobs that are a reflection of B factors from a crystal structure. So you have one side binding, the other side becoming highly dynamic. And this coincides with dynamics of water, egress of water. So in other words, high entropy. Okay, and if you go back to your early chemistry days, you think about things that are spontaneously driven. If you can ramp up entropy, you can drive a process forward. And you're losing entropy from binding a substrate, but you're gaining entropy from this other half of this machine becoming suddenly dynamic. And then there's chemistry that happens. You form a covalent. You have to do a so-called SN2 substitution. So let's just agree that there's some chemical steps that happen, some rearrangements. And then ultimately you form a covalent intermediate. And then finally that covalent intermediate that's temporarily stuck to the machine is then that it's finally severed, a bond is severed to release the fluoride ion and the other remaining half of the molecule through a process called hydrolysis. So there's another amino acid that breaks a water molecule and allows this molecule to be separated. So the point is not to talk about chemistry here, but the point is there are chemical events, there are dynamic events, and this is just a thermally driven machine, yet it has the capacity to sample specific functional states, right, in a specific sequence to achieve chemistry. And I have a cartoon. So this is not even an MD simulation. It's really just a cartoon that shows some of the functional states that this enzyme would sample. And if you think about it, there must be a functional state that the enzyme samples that allows it to trap or catch the substrate. Right? It's like if you play baseball, you have to be able to catch it. Before you catch the ball, you have to be able to open the glove. And so that's what's happening in this cartoon. There's an open glove state where the substrate binds, is desolvated, and falls into the pocket. Once it falls into the pocket, you'll notice it triggers dynamics that are actually faster. So I'm going to go back to this cartoon for a second and just point out, because I can't speak as fast as that movie. So you'll notice there are inherent dynamics in the molecule itself. Why? Well, they're thermally driven dynamic processes. And really what you're observing is the enzyme sampling the functional states that it needs to achieve the chemistry. Okay? So it's sampling those states, and specific functional states may be very short-lived until such time that the substrate binds. Okay? Once the substrate binds, which we'll see any minute now in this very exciting movie, the dynamics take off. The dynamics actually become, so the transition between functional states now becomes faster once the substrate has bound, and they become more pronounced. So, imagine it was sort of like a cartoon that Adnan showed this morning, a fuzzy blob. You have a fuzzy blob sampling dynamics, but now as soon as the substrate binds, there are restrictions in the pocket that now bias those dynamics to help achieve the chemistry. Okay? All right. Now, when I took biochemistry, it was a long time ago. But when I studied biochemistry, my professor taught me that proteins achieve their three-dimensional structure. They fold or achieve their three-dimensional structure. And part of that process involves sequestration of water, the expulsion of water. So the greasy or hydrophobic interior of the protein is hidden from the water which is expelled. So this makes it a spontaneous free energy driven process, right? Expulsion of water. And so that's more or less true, but it's also wrong. And you can tell from crystal structures, high resolution crystal structures of proteins, that of course the outside is coated by water, but you find a significant number of water molecules in the protein interior. Okay? A significant number of water molecules are found in a hydrophobic protein interior. Now, if you're a crystallographer from the early 2000s, you would conclude that the reason there are waters in these hydrophobic interiors is just to fill void volumes. But that's also not true. And what we believe is, in fact, many of these waters that are long-lived in hydrophobic interiors either hydrogen bond with themselves or with other parts of the protein, forming scaffolds. And these scaffolds are dynamic. As soon as a substrate binds, the scaffold can now rearrange via these water molecules and help guide the dynamics that take place in the protein. What a wonderful problem for allostery, right? It's not just about the atoms, not just about the amino acids, it's also about the water. Now, how would you prove that? How would you prove that waters contribute to allostery in proteins, in enzymes, in GPCRs, for example? Well, you could do it computationally using rigidity theory, as Adnan introduced this morning. You could also try and solve it, in part at least, experimentally. If I'm an experimentalist and I want to show that water is important for allostery, I first need to be able to measure some kind of allosteric process, some kind of dynamic process. And I can, and I'll show you how to do that. But then in a perfect world, I would want to change the Hamiltonian. I would want to change the energy of my water molecules. How could I do that? It turns out there's a convenient trick in chemistry. We replace H2O with D2O, so we take advantage of an isotope effect. Hydrogen bonds between D2O water molecules are about a couple of percent, 2 to 5% higher than H2O water molecule free energies, okay? But in a network of 10, I would now have 1.05 to the 10th power. I would have a significant measurable difference, right, in hydrogen bonds. And so this was the experiment that we tried. So how do we measure dynamics? What we do is this protein is full of tryptophan amino acids. So here you see a tryptophan analog with one difference. We've hidden a single fluorine atom in the tryptophan in place of a hydrogen atom. And if we do fluorine NMR, we can monitor all 9 tryptophans in this protein at once. And that's shown somewhere. I'll get to that in a minute. Okay, so I will show you, I'll give you a brief introduction to NMR, and I'll show you how we use fluorine NMR to monitor the allostery, to monitor dynamics of the enzyme. But the next question is water, tackling the water. And so as Adnan introduced, he uses rigidity theory to basically examine changes in degrees of freedom from such things as the binding of a substrate. So here in this cartoon, you see the binding of a substrate in one protomer and the changes in the degrees of freedom in the empty protomer, okay? And here he's mapped that as a function of amino acid residue, okay? It's from one amino acid to residue number 300. And you can perform this same experiment plus or minus substrate. You can also perform this experiment plus or as a function of removing the waters from the interior, putting the waters back, and seeing how that changes. So you have a direct computational way of evaluating these waters on the inside and whether they make a difference or not. You can also try MD simulations. That's also super useful because when you do crystallography of proteins, you can see the waters in the interior, but you don't know how they're arranged. Waters appear as dots, right? Because the only thing you're looking at are really the oxygen atoms, okay? And so you have to use computational biology or MD simulations to take over and arrange the waters that you can see from crystallography in the interior. And so the smart thing to do is start with molecular dynamics simulations to get a feeling for how the waters are organized in the interior. And this is mathematically a little bit challenging because how do you reference where the waters are when the protein is moving all over the place and jiggling? So the best way to do it is to do something that's sort of similar to the way satellites track people with cell phones, GPS, right? So if I imagine several reference points on a protein that each measure a distance to a water molecule, I can reference where my water molecule is in the protein without some kind of fixed coordinate system. And so this was done by Eugene and Sarah that are collaborators with us. And then then they went further. So Eugene and Sarah also went further to examine, with these starting structures of waters in the protein, to examine their configurations in H2O and in D2O. So not only does deuterated water have a stronger hydrogen bond with another deuterated water, but the potential functions are very easy to represent. So you can recapitulate that with MD simulations quite well. And what they found in their MD simulations were really interesting, for microseconds worth of MD simulations, they could find some kind of basin or ensemble of distinct different functional states, which was kind of cool. All right, so here's the money shot. So so I told you that these proteins have hydrophobic interiors, as do most proteins, and that you find, surprisingly, a fairly large number of water molecules in the hydrophobic interior. And many of those water molecules, as you can see in this graph, are very short-lived. Residence time nanoseconds. So this would be, a 100th of a nanosecond. So you find, within a 10th and a 100th of a nanosecond, very short-lived water. So they're percolating into the protein, but also running around and exchanging rapidly. But the surprising part is that there are a large number of waters, 40 or 50 per protomer, over here that have long residence times and also high occupancies. So that would be this upper corner here. Right? So even if one water molecule leaves, another water molecule fills its place, right? And so this is where we're most interested. These longer-lived, high occupancy waters, could they play a role in allostery? And what Eugene and Sarah found from their simulations was, in fact, a region in the protein where these long-lived waters resided. And by the way, it's a hydrophobic region, okay? And they formed these networks. And so indeed, this substantiates our hypotheses that waters could be playing a role in guiding the dynamics. And Adnan did exactly the same kind of evaluation using rigidity theory and found very corroborative observations. So the dark blue dots represent water molecules that have high occupancies and are long-lived. And they essentially form a dynamic bridge between the two substrate binding pockets in the enzyme. And D2O, by the way, has a slightly longer residency time than H2O. Okay. So now I have to torture you with NMR, but let's just imagine a scenario where you have a protein and you know that it's dynamic. And you know that, and you hypothesize that this protein could have specific inactive states. In this example, there are suggested to be 2 of them. And now I have a fluorine reporter. This is where my NMR signal would be coming from. And I put it on a specific part of a helix that I know is moving. Right? This is the part of the protein that I want to study. And then maybe there's some kind of intermediate state, and maybe there's an active state too. So these different configurations or conformations of this protein belong to what we call an ensemble, right? Different conformations. So this is the ensemble, and those different colored configurations represent specific states that we could talk about if we were pharmacologists. And then if we do fluorine NMR, this little reporter, this fluorinated reporter, will sit in different electrostatic environments for different states, and therefore we see a unique frequency or chemical shift, right? So 4 states, 4 peaks if we're lucky. And you notice in this cartoon that each of the peaks have a different width. And the width has something to do with how jiggly or locally dynamic each of those reporters actually is. So then here our reporter is a single fluorine atom on each of 9 tryptophans. And then if you look at the spectra, you can kind of make out 9 peaks. Here's a peak, 2 peaks here, another peak here, another here, here, and here. Okay, so these are our tryptophans. Different local electrostatic environments, unique frequencies. And you'll notice a few things. You'll notice this peak, for some reason, is broader than the others. It's wider. Okay, and you'll also notice really tiny peaks. Here are 2 small peaks. Here are 2 small peaks. What are those? Those are the other excited states of this protein. All right, so that it turns out that obviously upon binding of a substrate, the dimer becomes slightly asymmetric. And even without the substrate binding, you can see two excited state signatures here, one coming from one protomer, one coming from the other, that are slightly different. And this is an excited state, right? That's really a state that would be reinforced once the substrate binds, okay? And why is this peak so broad? It's so broad because there are local dynamics. It turns out that this peak is from a residue over here that's sitting right on top of the substrate in the active site pocket. It turns out to be broad because there are specific dynamics happening. Okay, so and then where do we go from there? Well, we can perform an experiment, an NMR experiment, that measures the transition between a ground state and an excited state, that measures allostery in a way that recapitulates a specific dynamic transition between one state and another needed to do the chemistry. Okay? And as Adnan said, many of these dynamic processes take place not on a microsecond time scale, but a millisecond time scale. And so they're very hard to recapitulate by MD simulations, but very easy to measure by NMR. And so I don't know if I should explain this to you or not, but let me give you an analogy. Many people do fluorescence spectroscopy, and often with a good fluorescent probe, we can measure a fluorescent lifetime. And the lifetime that we measure is a reflection of the dynamics, right? So I could have a very short lifetime if my fluorophore was bouncing around in some pocket, sampling different environments, and therefore serving as providing a mechanism for quenching or loss of fluorescence. So short lifetime, lots of dynamics. We can do better by NMR because we can create an excited state, just as you can excite a fluorophore with light. We can create an excited state, but then we can measure its lifetime as a function of refocusing pulses in an NMR sequence. So we can measure a lifetime with one refocusing pulse, 2, 4, 8, 100 refocusing pulses, get a different number, and measure a curve. So instead of one number, a lifetime, we can actually get a rate as a function of the spacing between the refocusing pulses. And so if we have an experimental curve like this, it becomes much more reliable to then fit that curve to a specific dynamic model. All right, so the dynamic model here would be this enzyme interconverting between its ground and its excited state. And so what I'm trying to show you in this data is that in water, in the absence of a substrate, we can barely make out any kind of dynamics. There's just a lot of noise, and it's very difficult to measure this transition. However, as soon as we add D2O, as soon as we go from H2O to D2O, suddenly those dynamics sharpen up, and we can measure this process to be taking place on the order of 2,000 times per second. Right? Bang, snap, put in D2O, you have this reinforced dynamic transition between these two states. And if you're a chemist, you may be surprised by this, because D2O, it turns out that deuterated water is more viscous than H2O. And so it's kind of surprising to see something speed up when you're adding something that's more viscous. When you're talking about a molecule here, and you're talking about things between atoms. And so the other question would be, what happens to the dynamics in the presence of a substrate analog? So we can add a substrate analog, which means it looks like a substrate, but the bond can't be broken. So we throw that in, and what you'd expect is if you throw in a substrate or a substrate analog, dynamics would take off, and they do. All right, so these are 3,000 in two measurements here at different sites, 3,000 and 4,500 times per second. And but what's interesting is that when we again replace H2O with D2O with substrate analog present, the dynamics become faster. Okay, so that's an experimentalist way of saying that water is important for allostery. Okay? Now, finally, you'd like to look at how efficient is the enzyme. So I've changed the strength of water interactions by going from H2O to D2O. Does this also result in faster catalysis? Because after all, this enzyme is supposed to break a carbon-fluorine bond. And so this is just math. It doesn't matter. But the point is that I have if my substrate is fluoroacetate and my product is fluoride, I should be able to see both by fluorine NMR, fluoroacetate, fluoride, and I can. These are thousands of spectra stacked on top of each other, but basically there's a time dependence where my product signal is growing, right? But my substrate signal is shrinking. And so here you see the areas under these curves. Product signal is growing in time, substrate signal is shrinking in time, and what I care about is the rate, right? I would like to know the rate, so I simply have to take the slope of each of these two curves, and that's you see here, and then I fit that slope to this messy mathematics, messy but easy mathematics, and then finally I have a curve that looks like this. So the way this works is you start with some amount of substrate. So here, 8 millimolars substrate. And I feed this to my enzyme, and then I monitor the rate of the reaction. And the rate of the reaction slows down because there's less and less and less substrate as a function of time, okay? Until finally I run out of substrate. And then I add another 8 millimolar, and now I'm back here, and I again measure the rate as a function of time, and it gets slower and slower and slower. This curve is higher than the next 8 millimolar aliquot because in the second condition here, I have product present, and the presence of product inhibits the enzyme. Okay? Now I repeat that entire experiment with D2O, with deuterated water, and you notice it's steeper, and this is a reflection of the fact that the catalysis is faster, right? So I see a decrease, right? The substrate is disappearing, the rates are slowing down. Then I add another aliquot, I'm back up here, and it starts to decrease again. And so the bottom line is that the catalytic rates differ by a factor of 2 by playing with the strength of the water-mediated hydrogen bonds. And, you know, I don't know if you care because you're AI people and mathematicians, but if you like proteins, the dimeric state is actually incredibly common. It is more common than a monomer in the protein world, okay? And half of Site's reactivity, which is the type of chemistry that this enzyme performs, in other words, you can only bind one substrate, you can't bind two substrates to this dimer. This is actually probably present in 1/3 to 1/2 of all dimers, of all dimeric enzymes. So it's also very, very common, probably for similar reasons. And if you have a dimer, you have cooperativity, you have allostery, and the two protomers have to have a way to talk to each other in order to contribute to chemistry, and this is allostery, okay? So it's a great, looking at a dimer is a great way of studying allostery. It's not the only way, but it's a great way. Okay. I promised you that I would get into some more practical things. I want to talk about nanobodies. I want to talk about drug discovery. So I'm just going to introduce you briefly to work that Adnan and I have been doing for the last 10 years or so on this really interesting family of membrane receptors. There are 800 of them in this family called GPCRs. You've heard of many of these. You've heard of opioids and therefore opioid receptors. You've heard of dopamine and therefore dopamine receptors. You've heard of serotonin, serotonin receptors, and et cetera, et cetera. So these are examples of receptors whose allostery we're very interested in for purposes of understanding mechanism, for purposes of designing new drugs or allosteric drugs, for purposes of designing, for example, allosteric nanobodies. And that is what I'll talk about. So I first want to talk about a concept that Adnan brought up this morning already, and that is this concept called promiscuity. So I mentioned to you earlier that a single human embryonic kidney cell contains 1 billion proteins, and every one of those proteins in the kidney cell talks to another 10 to 100 proteins, right? So there's this dynamic interactome, and GPCRs are no different. So here you see a GPCR cartoon, and what's shown underneath is its complexation with two different kinds of G proteins, the stimulatory G protein and inhibitory or G protein, also called GO. Right? So think about it. That's weird. If I had a puzzle piece, there's no way my puzzle piece would fit to two other completely different shaped puzzles. But in the protein world, you know, the protein is a dynamic machine, and it can sample configurations that allows it to engage one target and engage another target, even if they look different electrostatically and shape-wise. And what's curious is using the same fluorine NMR that I showed you earlier, we can actually observe two excited state peaks. Here's one that you see in blue and yellow, and here's another that you see in yellow. And the population of these excited state peaks varies depending on whether we add the cognate G protein, GS, or the non-cognate G protein, GO. And so if you saw data like this, you might guess that maybe there are two excited states, two activation states, and maybe the receptor has evolved this capacity to have two excited states in order that it can signal through one G protein and signal through another G protein. Identical agonist, identical ligand in both cases, but one of two states is more reinforced with the cognate or the non-cognate G protein. It gets even more interesting, but it has to do with pharmacology and maybe things that we can skip for another day. But the point is, these proteins are promiscuous, and so when you think about an allosteric pathway, it's even more complicated than that, because there can be allosteric pathways, as Adnan showed, for one G protein and then another G protein, right? And that's all embedded in this machine. Okay. I'll just skip this. This has to do with chemistry and how we make these machines. And Adnan showed this movie earlier today, but what the hell? So this cartoon is basically reminding you that this GPCR is very dynamic, and it's sampling these different functional states that belong to the ensemble, and that the role of the agonist that you just saw binding was not to bring together the G protein, but rather to activate coupling to the G protein and specific dynamic processes that allow for nucleotide exchange, dissociation, and downstream signaling. So it's, again, it's all about allostery. I'm gonna skip this in the interest of getting to good stuff. Okay, so in this cartoon, this again is and Adnan quickly showed you this morning. This is a cartoon of a complex between the GPCR, the receptor, and this G protein. Okay, and you'll notice I want to point out two things. This yellow pocket is where the agonist or activating ligand binds. This green pocket is where the nucleotide resides in the alpha subunit of the G protein. And upon binding of the agonist, this allosteric network or pathway is activated, and Adnan showed through rigidity theory that this allosteric network progresses through the beta subunit to the nucleotide pocket, right? So the dynamics are actually guided through specific regions of the protein predominantly that sort of allow for biasing of biasing of specific functional dynamics and therefore function. And that these allosteric pathways look different depending on whether the G protein is GS, the cognate G protein, or GO. And just to remind you as well, if you remove this chaperone, this beta gamma subunit, the allostery stops. You can still get a small amount of coupling to the G protein, so you can still get effectively signaling, but it's independent of the drug. No matter what drug you add, you have some very tiny output. In the presence of this chaperone, this dynamic chaperone, now you see very elegant allostery that depends on the nature of the drug that's added, be it inverse agonist, which shuts things down, partial agonist, or full agonist. Okay, I promised you something new. I'll skip all this. Here it goes. I'm going to show you another movie. Here we go. So this is an MD simulation. It's hard to do because this is an all-atom MD simulation for 5 microseconds for a receptor with thousands of atoms and thousands of water molecules and lipids and a G protein. And what you can see in this movie, is, I'll play it again for you, and I'll play the other one. So one is the cognate G protein, and one is the non-cognate. And what you'll notice is that from principal component analysis, the dynamics of the extracellular loops are very different. The dynamics in the NPX6Y in specific motifs in the receptor are very different. Dynamics in other motifs are very different in each of these two cases. Same agonist, same receptor, different G protein, and yet far, far away in these extracellular loops, the dynamics are different. And this is allostery. And if you were a pharmacologist and you saw this data, you might guess that if the dynamics are different, maybe you could design a drug that would work with one and not work with the other. All right. So even if the ground state structures look similar, maybe you could design a drug that would target one of these dynamic regions that are different between the two. And that's exactly what we did. And we did this using a transgenic animal. So Suda Sensei mentioned this morning about nanobodies. One way to make nanobodies is actually to take an antigen. So an antigen in this case would be your GPCR. Inject that antigen into an animal. The animal will make antibodies to that antigen. And you have to have a way of screening and pulling those antibodies out and then testing those antibodies, which we do. And a really convenient animal to use is not a rabbit, not a human, but rather a class of animals called camelids. And one of them is called an alpaca. You can make sweaters out of them. But another really convenient thing about camelids is that they make, instead of making massive IgG antibodies, they make these things called nanobodies that are on the order of 15 kilodaltons. And that's 10 times smaller than a big full-on IgG antibody. 10 times smaller means there's a good possibility that you can do something with this as an allosteric drug, okay? And so instead of working with alpacas, even though they're cute and you can make sweaters, another thing you can do is the same thing in a transgenic mouse. So one can employ transgenic mice that also make nanobodies, and you play the same game. And we did this with a company. And an animal will not make one nanobody. An animal will make 250,000 nanobodies. And so you have to have some way, and again, this is where AI helps. You have to have some way of examining or clustering, evaluating those different nanobodies and figuring out representative members and then testing them. And the shocking observation was that when we perform an activation assay of this target GPCR, we see an output that you see here in black arising from the agonist, okay, the endogenous agonist that's used on the GPCR. But then these new nanobodies perform really just as well. Think about that. Think about that for a second. Let's go back to this movie. So instead of targeting the active site pocket, we're targeting some loop. We're targeting some loop with a protein, and yet, you know, nature is giving you something back that's doing just as well as the drug itself, just as well as the endogenous agonist. And it gets better. If you combine the two, if you combine the agonist with the nanobody, you actually get an output that's even higher. And so I can tell you what we're trying now. Now we're trying to tether the two together. So we tether the drug through a PEG linker to a specific chemical group that allows us to attach it to the nanobody. So you would make a nanobody drug conjugate in order to do better at targeting and better at allostery and get cooperativity. And okay, so finally, do I have a few minutes left? Yes, I think so. Finally, I want to talk about fragment-based drug discovery, and I want to talk about it because, again, it exemplifies problems in allostery, opportunities in allostery, but also opportunities in AI. And so we have a project in Toronto with a colleague, Hui Peng, and the Structural Genomics Consortium. And we do what other people do. We start with a large library This library is available through a drug discovery consortium of diverse molecules, diverse small molecules. And we basically throw those diverse small molecules at the target, in this case, a GPCR, in the hopes that some will bind and will change the activity of the GPCR. And we've done that with very recently in the last month. And Huipeng has found about 6 molecules that seem to reinforce an active state and another 6 molecules that seem to reinforce an inactive state. So these would be allosteric modulators, okay? Promising. But at the same time, we wanted to design our own library for and utilize parallel chemical synthesizers from this library. And basically, if we can design our own library and make our own compounds, and then study how they bind, then on a parallel synthesizer, as soon as we get hits, we can iterate that using AI and make a new generation, test it again, make a new generation, test it again. So instead of having to buy your molecules from Enamine or use someone else's library, if you make your own library, you can evolve the species. You can put Lego pieces together in certain ways, make a new molecule quickly. And so this is what we wanted to do. And so if you're building your own library, the first question you will ask yourself as a chemist is, what must my scaffolding in the middle look like, right? What must my central scaffold look like? And we chose to build what we call cyclohexane heterocycles. So here you see an oxygen. So in these cyclohexane heterocycles, it could be oxygen, it could be sulfur, it could be nitrogen. And if you go back to your organic chemistry first class that you ever took, you'll remember these cyclohexanes can adopt chair-like configurations or boat configurations. And the point is that they rapidly isomerize. Now, then what we do is we decorate the cyclohexane with other chemical species, A and B. A and B would be good hydrogen bond donors, good hydrogen bond acceptors to make a drug, right? And it's all combinatorial. If you can make 100 different cyclohexane heterocycles, and then you add another 50 A's and 50 B's, you have 100 times 50 times 50. You can suddenly build a big library. And all of the molecules are non-flat. They're not benzene molecules. They have shape. And all of the molecules are stereoisomers, which means there's a mirror image of everything, right? And in a perfect world, what you hope is that one goes in and the other doesn't, right? And the other beautiful thing about a cyclohexane to a chemist is that if I now add a tag, so I can add, in addition to building this cyclohexane, I can add a reporter, a fluorinated reporter if I do fluorine NMR, and that fluorinated reporter will see the difference between a boat and a chair, right? So my fluorinated reporter will see a difference between axial and equatorial environments, for example, and the chemical shift differences as it goes from boat to chair or axial to equatorial are huge. If the chemical shift differences are huge, I can now more easily see by NMR binding versus not binding of my drug, which is what I want. Okay? And so it was partly for NMR reasons that we also chose this, but also for mass spec reasons. So we started building this library. And this is ongoing work. And then we wanted to try and test it. So we were specifically looking not for a drug for the GPCR, but an allosteric drug, Okay, and I'll get to what I mean by that momentarily. But just as you saw a nanobody bind an extracellular loop, here we're trying to find a small molecule drug that fits into a cavity or a crevice somewhere in the receptor and enhances the action of the agonist or attenuates the action of the agonist. If it enhances the action of the agonist, in our world we call that a PAM, a positive allosteric modulator. If it attenuates the action of the agonist, reduces coupling, we call that a NAM, a negative allosteric modulator. And here you see just a brief series of trial molecules that we've tried. And the way that we do this experiment is, again, we do fluorine NMR, and we do another silly pulse sequence that I won't tell you about. But the point is that if the signal drops when we add the protein, the target protein, this tells us that there's binding happening. Okay, for those of you that do NMR, it's a CPMG filtered fluorine NMR experiment. But the point is, if we see signal drop, then we know that there's some kind of binding. And there are different peaks here. because these represent different fragments. Okay? And I'll just point out this one in yellow at the bottom. So this molecule, 4%, the signal decreased by only 4%, so practically none at all in the presence of just detergent. In the presence of protein, my signal dropped by only 3%. So again, practically nothing in the presence of agonist. NECA is an agonist. But in the presence of inverse agonist, my signal dropped completely. It vanished. This means it's binding. This means, but it's only binding to the inverse agonist stabilized state. It's only binding to the inactive state of the protein. It's not binding to the active state of the protein. So as I said earlier, that makes it a NAM, a negative allosteric modulator. It's attenuating the action of the protein by reinforcing the inactive state. And it's that simple. It's that simple. And so skip this. So I guess, going back to a cartoon that Adnan showed earlier today, where he illustrated an allosteric pathway, if you think about it, now you have a way of chasing down or discovering allosteric drugs. You would take, for a given configuration, let's say, for example, a complex, a model of my complex between my receptor and its cognate G protein, you would essentially, using rigidity theory, you would examine an allosteric network or pathway, and then you would think about docking small molecules anywhere along this allosteric pathway and interfering with the allostery. Right now, we haven't done that. We've gone straight to experiment to test for allosteric drugs, but this is how one would start if you wanted to start computationally. You'd start with an allosteric network and then think about docking somewhere along that allosteric network to make your fragment work. Similarly, if, as I showed you in the movie, if you had some kind of extracellular loop that was also allosteric, then you could think about engaging it with a nanobody in a similar way, computationally or experimentally, to achieve something unique here. So allostery is alive and well, and I think there's a great opportunity with rigidity theory, for example, to help drive drug discovery of fragments, drug discovery of nanobodies, and just picking up on something that Suba Sensei mentioned this morning, something that I hope that maybe we can talk about today. AI can really help with regard to evolving fragments and fragment hits through multiple generations. So I'd be really interested in talking about some of your work in that area later today. Thank you very much for your time. Again, a great pleasure to be here.
Thanks, Scott. So you mentioned tryptophan labels and going deeper into the network. Mm-hmm. Is there a way to evolve that past tryptophans, or is that still challenging for other kind of residues to get more experimentally deeper readouts?
Okay. So, NMR people who do NMR are used to looking at everything, right? So you can take a protein like the enzyme that I showed you, or in principle the GPCR, and enrich that protein with an isotope, N15, and then look at every single amide in the protein. And that can easily be done with enzymes. It's not so easy to do with GPCRs. Or you could do another type of labeling would be methyl labeling. Methyl signals are strong in NMR, so you could isotopically enrich all of your aliphatic amino acids, isoleucine, leucine, valine, alanine, etc., with C13 methyl groups, and look at all of those, right? And so in both cases, you would have, instead of having a few tryptophans, like I showed you, you'd have hundreds of peaks. Then you would proceed through NMR strategies to assign those peaks, and then you would have what you're asking for, a lot of peaks. So for you, that would be great. For me, that would be expensive and long, but there's a compromise. Having dozens of reporters scattered throughout a GPCR or an enzyme is really useful to monitor, to get a fingerprint of different functional states and study dynamics, right? And I think in the modern world of high throughput, you want to think about something that's inexpensive and fast. And so I could talk to you offline about that, but things that have methyl groups in them, methionine is super useful. Tryptophans are super useful if you can invent ways to do methyltroses out of tryptophans. And so those are things that we're working on. But yeah, so for rigidity theory, you'd love to have atomistic resolution, but in the high throughput world, we're trying to think of ways of getting 20 or 30 reporters per protein to study dynamics.
Regarding the impact of the waters, so obviously those who are residing long time, they are important, but how about those who are short living? What is the importance of those? Are they important or?
Yeah, probably, you know, like the hydrophobic interior of a protein, it's not grease, it's sort of like methanol. So from a free energy perspective, if you have a dynamic machine, you're going to have water intercalating in different places. And almost certainly not every single water, short-lived waters, are probably not contributing to allostery. It's just a consequence of chemistry. However, the extent to which water can intercalate in the interior will dictate the hydrodynamic radius, the compaction, the packing of the protein. And perhaps many proteins have evolved not to be perfectly compact, but rather attain some sort of compactness that allows for dynamics. So the extent to which water intercalates may dictate the propensity or capacity of proteins to have a certain dynamic repertoire to move, right?
So technically, talking about the kind of contribution of AI into all of this, technically it could be viewed as some kind of combinatorial problem where we could, using the allostery analysis, kind of rank these water impact and contribution into those.
Yeah, I showed you that. I told you that story just because it's strange, right? But from a rigidity perspective, the waters have to be, they can't be neglected. The waters are part of the machine. They're certainly, as we showed, important to overall allostery. And Adnan and I are trying to expand on this work. I think you're part of that project too. We're looking at 1,000 more dimers, right, to try and get some kind of perspective of, like, in the universe of proteins, how important are waters in the hydrophobic interior with regard to allostery.
Thank you for your engaging talk. I am very interested in the nanobody with agonistic functions. I think, as far as I understand in your talk, I think this antibody, the nanobody, bind the loop structure where the small molecular ligand induces the structure changes, I think. I want to know how to generate, how to find this nanobody. Firstly, maybe you use some of the epitope area who was selecting the nanobody, or the accidentally you get this nanobody.
Yeah. So we didn't design it, although that can be done shockingly successfully. Yeah, that field is moving quickly. So we did not design it. Rather, we let the mouse do its work, right? So, and the beauty of that is if you design an antibody, you could run into a situation where you have a good binder, but a bad drug, right? Something that doesn't do well in the animal. If the animal selects for you nanobodies, you can guarantee that it's a good drug in terms of clearance, in terms of not falling apart in the animal, right? And then if you get a response from a screen, well, then it's also a good drug pharmacologically, right? So the animal can really help, but AI can really help. As soon as you start getting hits, you can feed that into AI to guide the design. So the two work together. And undoubtedly, many people are thinking about that. And that's the future. Yeah.
Okay, thank you.
Regarding the nanobodies, so I mean, obviously we have these mouse, but if you want to apply to humans, obviously we need to adapt some changes, and so in order to kind of, we have to kind of introduce the changes, right? And the AI could be like one of the options, and say these LLMs, another kind of structure-based methods could be kind of a nice option for the humanization.
But it's not as terrible as you think, because you're not introducing a mouse antigen into a mouse. You're introducing a human protein, a human antigen into a mouse, right? And so it'll make, sure, it has to be tested in humans eventually, right? But the mouse is making for you proteins that are meant to engage the human protein, the human antigen. So you're already ahead. Yeah.
So just one last question for me. Is it hard to make this kind of a nanobody together with that known drug? Is it like
A tether.
together?
A tether. It depends how good of a chemist you are. It was hard for us. It's hard to make it really efficient, but I think a chemist would answer that. I'm a physicist, really, but I think a chemist would say it's not that hard. It's known chemistry. I didn't talk about it, but you know, a protein, a nanobody, might have many cysteines. So how do you take a tethered drug and put it at the right cysteine on your protein? And there we took advantage of chemistry in the literature that allows us to attack just an N-terminal cysteine on the bottom of the nanobody, so it's tethered exactly where we want. So the answer to your question is it's completely doable, but multi-step chemistry, you do get losses with every step, and so you have to think about how to do that efficiently, and that can certainly be done.
And what's the motivation? Like why we need to
okay, great question.
Right.
Great question. So the target that we studied was the adenosine receptor, right? And so you'll find adenosine receptors in neutrophils. You'll for immune response. You'll find them in your heart. You'll find them in your brain. When you drink coffee, you know, the caffeine is an antagonist that blocks the sleep pathway in your brain. So the problem is adenosine receptors are everywhere. It's worse than that. They also have cousins. There's an A1 receptor, subtypes, A2 receptor, A2A, A2B, A3 receptor. So as a pharmacologist, you have adenosine receptors that look very similar and are everywhere in the body. So you have to make a drug that goes to the right cell and hits the right subtype. How do you do that? Because they all have the same active site pocket. You tether the drug to a nanobody that's specific to a given subtype, right? And then you could even go further. You could have that tethered to another nanobody that gives you specificity to a given tissue. So there are all kinds of problems that you don't think about, but can be solved, particularly with these conjugates. Yeah. Thank you for that question. It's a great question.
Can I ask something? Sorry. First, yes, about this library construction part. So I think you assume some kind of parallel synthesizer.
Yes.
And
yes.
Actually, I don't know about it. So what is parallel synthesizer? And, you know, how,
you have
capability?
We don't own one yet. I keep applying for them on grants and not succeeding. But in a parallel synthesis reactor, you could imagine if you have 100 cyclohexane scaffolds, you start with one scaffold and you put that in the reactor chamber. And now what you want to do is you want to add A and B, right? And you have 20 different A's and 20 different B's, right? So you'd basically, in one pot, you'd react it with 20 things, make 20 A conjugates, right? And then from there, you'd make 20 B conjugates. And so these reactors allow you to do more efficient chemistry and also separate groups of molecules that have been derivatized, right? And then you take those groups and throw all those fragments at a target and see which ones hit, et cetera. And the beauty of it is that it's you know, you choose chemistry that's easy. So you have a cyclohexane scaffold and you're adding things that are with the simplest of chemistry, A and B, to each of them, right? But then what happens is you get hits from 10,000, you get you know, 500 bad hits, 200 good hits, you know, 50 great hits. You take all that information, ideally with AI, and you ask AI what scaffolds work the best and what chemotypes work the best. Now tell me what my next generation should be, and then you repeat the process on a parallel reactor.
So this is very typical batch design problem. So but maybe to build an AI, then we need some kind of information about, you know, what kind of synthesizer we are assuming. So that's very good information. Thank you very much.
Yeah.
I don't know if this is a question, maybe a comment. So with this design of the nanobody, you can get really specific, like you mentioned. It can be sub-site specific because most of these allosteric sites are they're not so conserved, right? So that's the beauty of going after these allosteric sites. But I imagine with the presence of this conjugate linked with the nanobody, this will further stabilize the GPCR and the complex. So as we discussed with some our prior collaborators, that's probably the best way to prove where it actually is binding, right? What if we can get, like, say, cryostructure with the complex of the nanobody, then you really know if you're hitting those sites rather than just kind of hypothesizing.
Okay.
Right.
I don't know how to answer. I want to give you many answers to that question. I mean, first of all, like if I add an agonist to a receptor, in fact, it's still, as you know, it's still highly dynamic, right? I have an ensemble of different configurations. I now add an agonist, so it now biases the conformations to active-like states, of which there are many. So there's still inherent dynamics after I've added an agonist, right? But those active-like states are very important for the downstream coupling and chemistry, right? So, adding something doesn't necessarily make it rigid.
No, but with the present nanobody, then you can actually
and the nanobody is engaging we think, an extracellular loop.
Right. So cryo could,
which is also a dynamic region too. And in the early days, 4 years ago, when we thought about this work, we thought there was a part of us that didn't think it would ever work, because a nanobody is a big thing. It's not a small molecule. And you imagine a nanobody binding to this dynamic machine, this GPCR, and literally smothering the dynamics, right? It's not the case, though. There's a lot more finesse to it. You can have a big protein pinch a specific site and bias dynamics. Yeah.
And do you know which extracellular loop in particular? CO2 or?
Yeah, so with the adenosine receptor, there is no N-terminus. So you only have a small ECL1, a big ECL2 that through Adnan's rigidity theory we know is allosteric, and a small ECL3. And the nature of the experiments was such that the intracellular parts were not exposed. So we know the nanobody is binding on the outside. We don't know how or where yet, but we're certainly working on it. It's probably ECL2.
Thank you, Sensei, for giving me the opportunity to present at this workshop. So my background is not in AI, but in experimental protein engineering. For about 10 years, I've been collaborating with Tsuda Sensei by utilizing his machine learning model. We've significantly increased our probability of obtaining protein with the desired functions. So in my talk today, I would like to share more about our collaboration work. So Sensei and I met just as AIP was starting. So this is Dr. Kameda, who belonged to ICE, firstly connected us, and we teamed up with Dr. Saito, who is now a professor at Kisato University. So in our team, firstly, I experimentally create data, and then Saito Sensei adapt the Sensei's machine learning model for the biological application, and conducted the machine learning to obtain the protein with the desired function. Our research target on protein. Protein is a function molecule that is firstly generated from genetic information in the cell and major structure format for biologics. And this biologics market is worth $300 billion, and 90% of over 90% of biologics are protein-based therapeutics. The structure of protein is a linear polypeptide of 20 naturally occurring amino acids, and the function of protein is uniquely determined by their amino acid sequences. So, design a function of protein is simply about designing the amino acid sequences. So, this sentence sounds very easy, but actually finding amino acid sequences with a desired function is very actually difficult. Please imagine the protein with 300 amino acids. the number of sequence of combination called sequence space is much larger than the number of sands in the circular deserts. So, so finding amino sequences with desired function is so far more difficult than finding a single grain of sound in the circular deserts. Adding this challenge, the number of proteins which can be experimentally screening range from 100 to 10,000. So which is very much small. So therefore, finding a protein with a desired function is like wandering aimless in a map of these vast sequence spaces. So this is the reason why we developed the experimental technology assisted by AI to efficiently explore and find a novel protein sequence in these vast sequence spaces. So, against this background, this backdrop, we developed the experimental molecular evolution that integrated with AI. In this method, combo is utilized. Combo is a Bayesian optimization library developed by the laboratory, and our experiments combo was effectively applicable to a sequence space of 10 to the 6. So therefore, we developed this. Process to obtain a protein with the desired function. In this process, we first design a sequence space of 10 to the 6 by using experimental method or by dry techniques such as bioinformatics, protein language model, and generative AI. After that, we experimentally create training data and implement machine learning and conducted a combo to design a sequence space of 10 to 2. Finally, we screen the protein in the sequence space like that. So by using this method, we may constructively find the protein with a designed function. So this is outline of my talk today. And there are 3 topics. Firstly, I show you the potential of our approach to find optical variant in a desired sequence spaces. In these topics, we use the previous report for designing sequence spaces. After that, we apply the machine learning, that is combo for discovering the optimized variant in the designed sequence spaces. In the second topic, I show you the application of our approach to simultaneous multi-dimensional engineering for enzymes and antibodies. In these cases, I designed the sequence spaces by using the experimental approach and by informatics. In these cases, we show you the simultaneous optimization of function and properties of proteins. Finally, the topic is the application of machine learning to phase display. In this case, we use the next generation sequence resulted for training data. And then I show you the potential of the utility of the next generation sequences data. In the first topics, I show you the first success of our approaches. This experiment is the color change of green fluorescence protein to yellow fluorescence. When we compare the amino sequences of GFP and YFP, these 4 amino acids is different. So by changing this amino acid to this amino acid, we can change the fluorescence color from green to yellow. So changing only 4 amino acid residues sounded very easy. However, if we consider the sequence space sizes, we have to find the optimal combination of amino acid from the 160,000. So the challenge in this experiment is that can machine learning guide us to the sequence space of the best YFP among 160,000 variants. And second challenge is how many training data is necessary for machine learning to identify the best YFP. So in this case, I randomly mutated at only these 4 residues, so that we prepare this about 150 variant who get the training data. By using this 150 variant, we measured the fluorescence properties, yellowness and brightness, so that the many variant shows no fluorescence, and only 4 variants show the very weak yellow fluorescence. So therefore, this training data is not so good training data, but I applied this training data to the combo machine learning models. The combo machine learning model supplied the ranking of all the sequences according to the high yellow brightness. So that we prepared the top 60 variant, and then I analyzed the fluorescence properties of the top 60 variant. So that is many variants show the yellowness and high brightness. Especially, pretty sure that this is the top second variant. This second variant sequence is the same as Venus sequences. Venus sequence is very famous sequences which can show the highest fluorescence among these various fluorescent protein reported. Therefore, this result suggested that this machine learning approaches actually using is about the 100 training data with a few weak positive data can drastically promote the function of proteins like that is. This approach is this method I applied it to another protein, that is enzyme. SOTE. SOTE is a peptide transferase. In this case, I increased the sequence spaces. That is, we increased the number of amino acid residues position from 4 to 5. So that is we will explore the size of sequence spaces, that is 3.2 million. In the previous reported, these 5M variant have been reported, which is more activity than wild type like that is. In this variant, these 5 positions are mutated to a specific amino acid, so that this is a 5 variant shows the more activity, more catalytic activity than wild type like that is. So in this case, we randomly mutate these five amino acid residues, and then we prepare these about the 8 variant for training data. So the size of training data is only the 0.03% of sequence spaces of 3.2 million. So that is we obtain this training data where there's many variants show these less activity than wild type, and only 3% variants show these more activity than wild type like that. However, when we use this training data and machine learning and prepare these top 50 variant predicted by machine learning, the predicted variant shows the more activity than wild type like that is. So in this case, we use the 2 type of training data. One type training data is this is 82 variant extracted. Another training data is that we added this is 5M variant like that. This 5M plus. So this result is this result was from the training data with 5M variant. This training data from this result is training data with that is that is 5M variant. So actually, we in this case we try again to use this data, the training data to, and then we apply the machine learning again. So that is we find many high, much more activities than wild type like that. So many variants show the more activity than this 5M variant like that. When we compare these two types of machine learning, so that is we same high activity variant between these utilization of training data containing 5M variant and without 5M variant. 5M variant. Therefore, our result is independent of the using of the high activity of 5M variants. Next, I show you we visualize where we research in sequence spaces by using principal component analysis. So in this sequence spaces, spaces by made by principal component analysis, the position of wild type here and the position of 5M variant here. And the training data is plotted as this is a grade plotted like that is. When we plotted is the first result is here. So this is the data of the machine learning guided library with the data 5M variant. In the blue plot is represent and the data presented the variant of machine learning guided library with the data of 5 variant. So by using the data of 5 variants, drastically drastically changes the predicted sequences like that. So by using these 5 variants, machine learning predicted pre-researches around the 5 variant like that. Next, I plotted the top 3 variants in each libraries. So this plot indicated is in this sequence spaces there are two main mount, but the position of 5M variant and wild type variant is not the top of this mountain. So maybe the top of mountain existing around here maybe. So next further we plot is the second result of machine learning here. So further this result shows the top of the mountain is around here, not here, and in this here this is the top maybe here and here. So in this this result indicated that we can improve the protein with only 100 training data with a few percent positive data, and but if we change the kind of positive data, we can change the pathway to go to the molecular evolution. Next, I move to the next topic, that is the application to simultaneous multidimensional engineering for enzyme and antibodies. Key feature of our method is using of only 100 quality training data set. And this feature enables us to obtain multiple function and properties for training data. So this this thing lead us to this idea. Can machine learning guide us the sequence with several properties simultaneously optimized? Here I show you the trade-off relationship between function and stability. this situation often happened in mutagenesis into protein. Mutation to increase characteristic and mutation to increase target affinity often causes the destabilization of structural stability. So when we increase when we try to increase the catalytic activity by mutagenesis, this mutagenesis often causes the decreasing of the structure stability like that. In the same situation, we also show you in the case of antibodies, when we try to improve the binding affinity of antibody like that, this mutation often causes decreasing of structural stability like that. So therefore, the challenge in these topics is that can machine learning guide us to simultaneous optimization of function and stability? So this is, I show you the result of here I show you the result of simultaneous improvement of expression, heat resistance, and alkali resistance. In this case, I designed the first sequence spaces by experimental approaches. Firstly, I performed the airborne PCR and I performed the plate assays. And after that, I decided the three positions where should be mutated. At these three positions, we randomly mutated, and I prepared about the 250 variant. I measured the expression in E. coli and heat resistant and alkali resistant. So that this variant showed the only 0.5% of 250 variant showed the positive data. So therefore, this many variant showed the negative data. However, we applied this plain data to machine learning, and I prepared about the top 50 variant predicted by machine learning, so that we found the many positive variants like that. So many variants expressed in E. coli at about the 2 or 3-fold of the wild type, and many variants show the high resistances against alkali and heating. So important thing, this top variant show about express the 5-fold expression level and the 7-fold of heat resistance and alkali resistances. For the variant with the in this slide, I analyzed more quantitative properties of the predicted variances for variety. So that is when we compare the expression in E. coli, this wild type protein do not express it in this strain of E. coli. However, another variant predicted by machine learning showed the expression in this bacteria. And then this the number of the variants showed the heat resistant. So when we heated this protein at 6 degrees, after that we analyzed the catalytic activity. Wild type showed the no activity, but this is a variant showed the comparable activity than the protein without heat treatment. In the resistant pH, this in the treatment of the solution at pH 10, wild type was showed the no activity, but this variant showed the the catalytic activity. So that is by using this our approaches, I could obtain this variant which can highly expressed in E. coli and show the resistance against the pH and heat treatment. Next, I show you the anti-COVID COVID VHH antibody nanobody. So in this case, I also decided the sequence spaces by means of experimental approaches. In this case, I conducted the error-prone PCR and saturation mutagenesis for this antibody fragment, and then I performed a phase display separation process and then we analyzed the data by using the next generation sequences. So that we decided the 5 positions should be mutated. So that after decided the first sequence spaces, we randomly mutated these 5 positions, and then we created a training data for binding, thermal stability, and expression. After that data, we applied this training data to machine learning, and then we prepare top 50 variant for analyzing the binding, thermal stability, expression. So that is we obtained a very good this variant. So these 3 variants show about the 5-fold show the high expression level at the 5-fold of the wild type, and then this variant shows the high thermal stability like that is, and then all variants show about the 5 times stronger binding activity than the wild type is. Further, we applied this our approach to the improvement of cytotoxicity and expression. Here, I designed this bispecific and bivalent antibody we called Vivian. This Vivian, I applied this is the antigen binding fragment with affinity for the CD3 on lymphocyte, and then we applied this VHH fragment with affinity for the HER2 cancer antigens. By using this fragment, I designed this bispecific and bivalent antibody. In this case, I mutated only this VHH fragment to change the binding affinity to the cancer cell, and then we make a training data for the cytotoxicity and expression, and then I conducted this machine learning process, and then I again prepared about the top 100 variant to analyze the cytotoxicity and expression. This is training data, and this is the data from the variant predicted by machine learning. So in the training data, many variants shows the less expression level and the less cytotoxicity. However, by using using the training data, the machine learning predicted many higher expression and higher cytotoxicity like that. So these approaches can use it to the improvement of cytotoxicity and expression simultaneously. In the next, I show you the application utilization of informatics to design the first sequence spaces. So in this cases, I show you in this cases, I use the consensus engineering for the designing the first sequence spaces. In this case, we consider the most frequent amino acid in alignment of homologs. Immunoglobulin variant dominant is the most most favorable amino acid for some dynamic stability. So by using this concept, we decided the position where the used amino acid is not used in the alignment should be mutated. So in this case, we mutated about 20 amino acid reduced positions, and then we prepared about 19 variants. I analyzed the expression in E. coli, expression level in E. coli, and binding affinity by means of ELISA, and then relative stability by means of ELISA. By means of ELISA, like that. In the training data, I think many variants show these around these proteins around this wild type. However, the machine learning, the variant predicted by machine learning show this to hold shows the higher expression level and higher stability than wild type, like that, is without loss of binding affinity, like that. So machine learning predicted an SCFB fragment with 2-fold better stability and 4-fold better expression at no cost of affinity. So in this result, we get that this is a good FB fragment information. Therefore, I graphed that this is FB fragment into the full length IgG antibodies. So antibody, when we hit this IgG type full length antibodies at 60 degrees, the wild type antibodies are aggregated at for 20 hours. So this plant antibody are aggregated at heating 60 degrees for 20 hours. However, the antibody antibody where the predicted FV fragment is grafted, so this is no aggregation. Even at the heat treatment 60 degrees for 12 hours. And then this antibody shows the same binding affinity, and further the expression level in mammalian cell is we succeeded the increases at 3-fold of the wild type on parent antibodies. Now I applied this application to the expression and solubility. Recently, solubility of antibody is very important because by increasing the solubility, we can prepare the high concentrated antibody therapeutic solution. So that actually we succeeded in the improvement in the solubility like that by using this method, and then we increased also the expression of IgG in mammalian cell without loss of binding, like that is. This is the final. So this is the final application of this our method. This is a humanization of antibody fragmented. In this case, I try to simultaneous optimization of humanization, expression, stability, and binding affinity. Also, the improvement of humanization induces a little bit decrease of the expression, but we could keep stability, and we could improve the affinity, like that. So our machine learning predicts variant shows the improvement of humanization with the loss of stability and binding affinity. So next I talk about the final topics. That is application of machine learning to phage display. What we talked so far is about this application of our approach to lead optimization, where function protein is improved. In our trial of this process, we succeeded in creating 100 training data containing a few percent of positive variant. However, in the process of expressing function in non-function protein, that is lead discovery, creating only 100 variant would not allow us to obtain data containing even a few percent of positive protein. So from here, I show the iteration of next generation sequence data to obtaining antibody and antibody mimics. So from here, so in this topic, I applied the phase display method to obtain antibody with affinity for a specific targeted. In this case, we prepare a large number of variants. That is the library size is about 10 to the 10th. And then we displayed the variant on the surface of file display, and then we performed the selection process, that is panning selection like that is. By repeating this process, we can select the binding antibody or displaying on phase display. However, in some cases, these positive variants are not always selecting because in this process is very complicated. Therefore, a little difficult to selecting the phases displaying the antigen binding antibody fragment only. So therefore, this challenge in this topic is that can machine learning discover more function variant than the variants experimentally identified? And can machine learning discover function variant with the data? There are no positive variant are experimentally identified. So firstly, so for this experiment, I applied this data. So here I randomly mutated only the CDR loop region. The sequence spaces size is about this is 10 to the 25. And then we conducted the first display selection process, and then finally I obtaining this antibody VHH antibody fragment with low affinity at about 600 nanomolar. In these cases, I applied the NGS analysis at each sub-library in this selection process, and then I try to use this NGS data for machine learning process. Important thing in this process, we cannot get this experimental binding data because we can get many antibody fragment sequence information, but it's very difficult to separate that this is separated monoclonal antibody fragment and analyze the binding data. Therefore, here we consider this in the NGS analysis, the binding number maybe has a relationship with the binding affinity. So therefore, we make it the indirectly linked data between sequence and binding strength. So we cannot directly analyzing the binding strength of each antibody fragment. So we cannot connect the directly linked data between sequence and binding function. Therefore, instead of the experimental binding function data, we applied this lead number instead of the experimental binding affinity measures data. So by using data, this is the result of the NGS data from the second round of file display panning results. So by using this data, I use this data, training data to machine learning, and then we selected the top 10 amino sequences predicted by machine learning. I show you that this is the probability of amino acid at each position. So when we compare these are sequence logo, I think by using machine learning, we can select the specific amino acid at each position. So that we, according to this data, we try to prepare this machine learning guided library as a second library, and then I apply this library to phase display process. After that, after phase display using this second library, we separated this about 40 variant and analyzing the binding by a monoclonal phase ELISA. So that we found several variants which shows the higher binding affinity than the variant which found only experimental promotion. So that we succeeded in obtaining this antibody fragment with higher affinity than the antibody which was found only in experimental process. Therefore, deep sequence and machine learning discovered higher affinity antibody fragment than wild type. So final trial, I show you the application of no positive the phase display library information without positive variant. In this case, I applied these uses the antibody mimics where about the 11 amino acid positions are randomized. So in this case, I also perform this phase display system at four round, so that maybe this library and maybe we could succeed in some binding some of the positive variant in the sub-library at second, third, fourth. However, we separated about 100 variant from the second round sub-libraries, but we couldn't find the good variant with structure for this. For example, this variant shows these many morphology of structure morphology like that. So important thing, this antibody, this selected variant shows the aggregate forms like that. Further, this variant shows this non-specific binding for protein, and this fragment has no secondary structure like that. By using this data, I tried to find some antibody mimic fragment with affinity who was targeted. So I also applied these deep sequences, NGS data as a training data, and then we selected the top 100,000 variant information. After that, we crystalline to separate to the group of the 9 groups like that is. So when we see these 4 groups, containing is about top 1,000 variant in this library. Therefore, we try to prepare the secondary library using this information of this clustering. So that is we succeeded in getting several 4 variants which show the specific binding to the targeted. this variant shows the monometric forms and then show the native wild type like the second structure, and then these show the selective affinity binding for the specific target. We further analyzed the binding affinity by means of ELISA. So these variants show this this is EC50 of about 100 nanomolar. Therefore, by using this method, we can mine some functional variant from the not good is the file display result. In conclusion, the machine learning can guide us to the optimal and suboptimal variant if appropriate sequence is designed. Machine learning can guide us to the variant where match properties are optimized. And informatics may be useful for designing a sequence space where machine learning can discover the optimal and suboptimal variants. Finally, indirectly linked data between sequence and function data prepared from the NGS may be used for mining function antibody. This is acknowledged. That's all. Thanks for your attention.
Yeah, thank you for your presentation. So I'm curious, like what machine learning model you used?
I use a combo.
Okay, and for like multi-objective part, so there are like conflicting objectives.
Yeah.
So like what sort of model you use?
Actually, I applied the combo, but the combo is not the multi-element. Therefore, when we consider the scoring, I joined the two information, the binding and some stability, and then we make it one data set. So that is therefore I changed the two different information to the total one information to making a machine learning for a combo application, combo model.
Okay, and final question is like, so in your this scoring function, did you consider any sort of like diversity or novelty kind of metric to? So I see in the result that there are some like known, which are not known to the training data. So you have some novel discoveries. So just by using that scoring, that result you got, or you used some sort of like other component so that your machine learning model can predict beyond the training data. So this is your known region, like the training data. And if I look at the results, so there are some novel region, right? So in that novel region part, so you have used some sort of scoring. So apart from that scoring, did you use some sort of like diversity or some novelty based guidance so that you can go beyond this one region?
Actually, I think you said that when we compare the training data and the predicted variant, when we compare that in the same ranges, I think is this is a good or not. I think of course I did that. So that actually when we compare the scoring, of course the variant predicted by machine learning showed the high scoring than the variant used in training data.
So I mean, as far as I know, it's just a Bayesian optimization without any modification. So
okay.
So no such fancy. Yeah.
Umetsu Sensei, thank you for your talk. Kind of, I'm interested just kind of from your practical insights. So there was always like, you're not considering whole search space, but you're always considering like 5, 11, but just like, how do you like choose what's the like practical solution? You have like maybe some kind of a known binder, and then you just randomly mutate some positions and some decrease affinity, some kind of maintain, and then based on that, or there are some other, how is it performed?
Sorry, one more time. Sorry.
No, so I mean, like you have a nanobody, okay? So that search
Mm.
code search space is like a 20 power, I don't know, maybe, I don't know, 100, right? But in the end, like you end up with like 20, with like 5 or 11 positions, right? In the end, so you not all positions are considered, but just several, right? So how are you selecting them?
In this case?
Yeah, for example.
Oh, yeah. You mean this? Now in this case, I specifically mutated this only 5 position. But because I find it, I could identify this experimental session. I think you mean that if we can increase the position of.
Yeah. So
Okay.
why is these 5 not to like, because they are probably located in those CDR3
yeah.
loop, but maybe like something in the frame. I don't know.
In this case, in this case, when we use combo, we have to design the sequence space size to the 10 to the 6. Therefore, we have to decide on 5 positions in this cases. But here, I think in this case, we of course sometimes it's very difficult to identify the 5 positions. In these cases, we often approach this with of course the candidate of mutagenesis position is maybe the more than 5 positions. But when we consider the exploring sequence spaces sizes, so when we research, when we explore the amino sequences where only 4 mutated in these about 10 amino acid residues. Okay. For example, 10C4, 10C5. By using this process, we apply these other approaches.
Okay, then, yeah.
So if you start with an error-prone PCR step and you find, let's say you have an example where you find 20 residues across a 300 residue protein that seem to be important, that bias some of the properties that you want to optimize. Where do you go with 20? Like, I can understand 5. 5 is perfect because you can generate a controllable number of variants. But like, I could imagine an error-prone PCR getting 20 hits that seem to be indicative of some of the parameters you want to, some of the properties you want to optimize. So where would you go after that?
Sorry, one more time. Okay.
Yeah, you had a slide you were just showing Andreas just prior to this one.
Fine. Maybe I think your experimental section is okay. Just a moment.
It was the error-prone PCR step.
Yeah. Just a moment. Here.
Yep. Yep. So instead of, for example, 5 positions, suppose that were 25.
Yeah. I show you that, and another slide is, and this is evidence how to decide the 5 positions. In the case of saturation mutagenesis, I applied the saturation mutagenesis at each position in CDR, only CDR. And I mix this library in the phase this way and perform the panning procedure. So that is after panning, we analyze the NGS analysis. So that is we analyze the percent of mutagenesis, and then we decide the high percent of mutagenesis we should apply the mutagenesis to increase the affinity. In the case error-prone library, we applied this error-prone library throughout the whole protein, so that we found some good position. Some position should be mutated at the framework region. So that is we decided that this 3 with 5 positions by using this method.
So what?
Okay, thank you. That answers my question. in the case of the sortase, do you ever try and, is there an assay for the sortase that allows you to test survivability, because then you can keep evolving as opposed to making separate species and testing them? Do you know what I mean?
In this case, is you mean in this case, yes, so this, yeah,
Right.
This
So
case is, I identified the position mutated according to the previously reported.
okay.
Okay, and then in this case, it is sorry, one more time question. Sorry.
Yeah, I guess so. That's you're not making 20 to the 5th. Are you making 20? You're not separately making 20 to the 5th sortases, right?
What, 20?
Well, you have 1, 2, 3, 4, 5
Five
residues of
position.
interest, right?
Yeah.
But do you make 20 to the 5th species? No. I guess your training data, I guess you have it there.
25th?
Um,
Actually, I prepared variant 82 variant.
82. Okay.
Randomly mutated.
Okay.
Simultaneously. Yeah.
All right. So then classically, I guess, if for example, sortase, when reacted with a substrate, produced something that gave survivability to the cell, then you could make an infinite number where you do error-prone PCR on those 5 sites, right?
I don't apply the airborne PCR.
Okay.
I apply specific method
Okay.
at only this position. Yeah.
Okay. May I ask one more question? Yeah. Do you ever look at, like, once you find hotspots, do you ever try and make pairwise variants? So K190 and D160. You try all possible variants of K190 in combination with D160 to look for correlations to guide you into ultimately a very big sequence space that might be more productive. Cooperativity. Do you ever do any?
Or epistasis.
Okay.
It also maybe related to epistasis, right? So like epistasis, right? So like with these fluorescent proteins. So how do you deal with that? Do you have this epistasis effects, like just from the looking at the experimental data?
Well, I'm not sure, but the question is, so actually you can take 2 positions and try all 400 mutants. So maybe you did this saturation analysis for 1 position.
No, I don't
But
apply.
you may do this for 2 positions, but you didn't, right?
Yeah. Only simultaneous simultaneous mutation for the 5 positions. And then therefore I don't do the specific the mutation for 1 or 2 only. I don't do.
Okay, so time is almost up. So thank you very much for the discussion.
Okay. Good afternoon, everyone. My name is Masanobu Naito from National Institute of Material Science, NIMS. So first of all, I'd like to express my special thanks for Tsuda Sensei for giving us this great opportunity to present our recent work. So the topics I'm going to talk about today is mainly focused on the polymer informatics. So now my expertise there is polymer development. So by using the machine learning. So before starting my talk, so first let me briefly introduce my institute, National Institute of Material Science. So here is the center of Tokyo. So our institute is located in the part of north from here, about 1 hour by trains. So if you go to Haneda, so that you can take a bus or shuttle bus or the train, so it takes about 1 hour. So if you have a chance to please come to visit our institute. So this slide shows our representative research achievement. So everyone has the cell phone, right? So if you have the cell phone, so everyone use our materials, so called the phosphate. So this is a red emit phosphate. So this is invented by our institute. And also guys bought the airplanes, Boeing 787. So you can see the Rolls-Royce jet engines. That is invented by NIMS braid, the high thermal resistance super alloys. So that is our materials. So the as shown here, our institute specialized material development, only the materials. And then recently we covered all of materials like electric energy, magnetic structural materials, the polymeric materials, as well as the biomaterials. And also our important mission is to maintain the shared facilities. So we have the world-class cutting-edge facilities in Japan. And this is our very important initiative of the so-called the M-cube, material M-cubes. So that stands for the MOP, so that is the materials open platforms. So that is kind of the open platform system to share their facilities and the collaborative, provide the collaborative circumstance for the both industrial and also the academic. And also we have the material global centers. So that is kind of the research center to exchange the researchers and the students that we accepted the students from all over the world. And also the, yes, we collaborate with the international collaboration with the many countries. And also the most, today's, the most related part is here, material research bank. So we have the large material research data banks. So that is creating, our mission is the creation of material data platforms. So we collect the curated data, we generate the data, the keyword data, and also the using the data. So that is our big missions. And also develop the knowledge network, like including the AI systems, materials informatics as well. And also we have the common facilities to world-class measurement facilities. So that is the very important initiative, the so-called materials M-cubes project. So within this context, we have we are eager to initiate, we are starting to do the material DX platforms to accelerate the innovation of material development. So that is has the three main pillars, main components. One is the DICE, that is the sort of the database of the materials. So maybe some of you may know the PolyInfo. That is the biggest polymer databases. So these data was prepared by hand pickings. So the researcher read the papers and put the data, all data into the databases. So we keep these activities for more than 20 years. So that is we have now we have more than 300,000 data set, data point. So that is the world largest big database of the polymers. And also we have the AtomWorks, that is for inorganic materials. And then, and also recently we're focusing on to develop the self-driving laboratories, so to generate automated generated the data. And then generate the data and keyword the data, store the data into the database. And also we have another AI assisted database, so that's so-called RDE, Research Data Express. That is so it's not on, it's focused on it is specialized for the machine learning. So if you put the data, so they automatically structure the data. So if we put the data, metadata, the raw data, and also the invoice, they just get together, put in the database. So they automatically structure the data. So and also you can use these data. And then also finally, the most important thing is how we utilize those data. So now we are eager to develop new materials by using those infrastructures. So maybe I already explained about this. So the the 800,000 data entries already. Yeah, the day by day. So that we increase the data and the data, and also the inorganic material, something like that. So basically this is the freely access to these database, but some of database is the paid service, so that you can, if you paid the money, so that you can get more data. So that is, I think that is reasonable. And then here the important things. So that now we can access to the database of the polymers. Okay, so by using this database, but how we perform the machine learning, that is important things. So so we have to express the molecules by somehow, like by using the smiles, the whatever graph, the moles, something like that. So that is sometimes cause a problem. So because there is no perfect descriptors to express their molecular structure, express the physical properties of the polymers by using such kind of descriptors. So another important thing is the polymer property of the polymer is not easy to understand. So the primary structure doesn't express, cannot predict the molecular structures. So the more higher structure is sometimes important to express the properties. So this graph shows, this slide shows that this is primary structures. So that you can, of course, you can describe these molecules by chemical structures, but that is not sometimes not mean. But you have to think about their tacticities and the sequence, and also the network structure, something. And also the more higher structure is more important for example, the mechanical properties like that. So in that cases, so not only the structures, but also the process is also important to how you process how do you make it this kind of the polymers. So that is also important. So you have to understand the molecular structures as well as the process informations. So that's quite difficult, the things to perform the polymer informatics. And also if you want to clearly understand the such kind of properties, so you have to analyze by using bunch of the operators. So if you want to see the surface structures, you have to do the AFM, the AMM, blah, blah, blah. And also the NMR is a great information about the structures, conformational information and the structures. But of course, that is very important. But especially in the case of polymers, so if you want to perform if you perform NMR and also the GPC, gel permeation chromatographies, so at least polymer have to be dissolved in the solvent, something solvent. Solution is necessary. Of course, the solid state NMR is available, but to more easier, so the solution phase is the more easy to understand. But that is quite difficult. And also another big issue of the analysis of the polymer is there is no polymer sequencer for artificial polymers, synthesized polymers. Okay, so that is quite important. So the limitations. So how you understand the polymer properties by because you cannot tell the sequence by NMR. So that is only limited cases. So you can tell the structures. But the good example is today's topics, the proteins, the biopolymers. So you can tell the three-dimensional structures, folding structures, and then by using the NMR, the X-ray, something like that. And also you can tell the sequence, because you can tell the sequence of the structures. Also the monomer species is limited. So only the 20 amino acid participated into the making these polymers. So that is the point of that's one example. Then, so very classical method to understand the sequence of the biopolymers. So that is for example, the DNA or the protein. So the first fragmented the polymers, biopolymers, then make the fragmented by the certain rules, like an enzyme, something. And then after that, reconstructed to decode the structures or to understand the original structures. So that is the conventional method. However, so in the case of the synthesis polymers, there is no fragmentation method at a certain point. That is a big problem. And also the important thing is recently, and not only the biopolymers, so the synthesis polymers, also the sequence is very important, so the discovery. So for example, in the case of the proton transport channels, this is a mimic of the proton transport, the proteins, something like that. So they use the amphiphilic polymers, hydrophilic, hydrophobic polymers exist in the polymers. So in this case, very short segment, like a hydrophobic, hydrophilic, that is sort of the very short segment, short sequence, significantly affected to the proton transport proteins. So, and also another example is the thermal conductivities. So also these polymers as a copolymer of polypropylene, polyethylene, copolymer of the polypropylene, polyethylene. That is not real. That is only the computational studies. But so this copolymer with the certain sequence shows the very high thermal conductivities. That is predicted. So surprisingly, this paper is very highly cited, even though the polymer is not the realities. So that is quite interesting. But the important message is the short segment, the sequence is very important to express the function of the polymers. That is very important. So we have to understand the sequence of the polymers, of the synthetic polymers. So how do we do that? So as I told you, so that we cannot cut fragmented at a certain point. But so here we use the machine learning method. First, the method is like this. So first we randomly fragmented it. So just cut the polymer species to make a bunch of the big databases. And then after that, and classified it with machine learning to decode it, the unknown random copolymers. So that is our strategy. That is totally reverse the chemistry, reverse method. So how do we do that? So first we have to randomly decompose the polymers. So that we just use the high sensitive mass analysis, so-called direct analysis real-time mass spectroscopy, DART mass spectroscopy. That is quite simple method. That is the atmosphere mass spectroscopy. You don't need the high vacuums condition. And also just put the samples here. You can take samples, polymer or the organic polymers like this, and then to put in the sample here. So from here to here, so they ionize the plasma jet generated to ionize the materials. Then the ionated materials can be detected by the mass spectroscopies. That's quite simple. The advantage of this method is, so you don't need just the solution. You can, even though the polymer is insoluble, and also the composite materials with the polymeric materials and inorganic material composite materials, so that you can use it. So as long as you can just heat it, samples. So that is a very convenient method. So for example, so this is a polypropylene, commercially available polypropylene data. So with different levels. So you can see, so okay, there is many polypropylenes. Okay, so the PET bottles, the cap is polypropylene. So however, there is molecular distributions are different, and also the molecular ranks is different. Okay, and also the additives, so they also different depending on the companies. Okay, so so if you look at the polypropylene database as a labels in the labels, but the materials are totally different. Molecular weight, molecular distribution, and also the additives, totally different materials, just a composite materials. And also, so that this method can tell which polymers identified. However, the only problem is the mass spectroscopy cannot tell it's not a quantitative. So the mass spectroscopy cannot tell how much the signal is there. So that is a qualitative. It's not a quantitative data. So how do we, so to analyze the more precisely the amount of the fragmented components, you have to quantify those data. So how do we do that? So I'll let you know later. So the first, we have to get the big data. Okay, so how do we do that? So of course we use the that mass spectroscopies, but that is not so easy to make the big data. So to enhance, to increase the number of data, so we use the pyrolyzers. So in that case, so we put the samples here, only several micrograms of the samples, and then heat it to pyrolyze these samples. And then only the gases, phases, the fragmented molecules can detect it by the mass spectroscopies. So by changing the temperature of these heaters, so we can obtain the heat map data at once. Only within the 15 minutes, we can obtain the big data of the mass spectroscopies. So each temperature has the mass spectroscopies from the room temperature to 600 degrees in Celsius. So we can get within the sweeping the temperatures so that we can obtain the many data at once. Okay, so by using that mass spectroscopy, so the first we fragmented the polymers to get the big data, pool of the fragmented polymers. And then, so we use the unsupervised machine learning techniques, so-called maybe the next slide, I'll tell you, the NMF method to classify the data. It's a non-supervised data. And here's very important. So first, this is unknown random copolymers. Okay, so we don't know the sequence. Okay, and then fragmented by heating, cutting, chopping the data, the main chain of the polymers, and then make a fragment of pool of the fragment, and then so by using the supervised machine learning, so you can tell which spectra is corresponding to this short species. For example, in the case of the blue, green, blue, green, whatever. So then we calculated the number of this segment. So that we can calculate how much this segment containing this mass spectroscopy. So we can tell that. So this is imagine. So this is sort of the alternative copolymers. We can think about this is the kind of the well-defined alternative polymers. Blue, green, blue, green, something like that. So if you know the length of these polymers, so you can tell how much these components contain this original random copolymers. We can tell that. So that is our strategies. So to analyze, to enable the method, we use the NMS method, non-negative matrix fluctuation method. So that this is the kind of the image analysis, data analysis, image analysis method, conventionally, the image analysis method to reduce the size of the data. Okay, so this is original the heat map data. So by using the animal treatment, so we get the basis vectors. We can obtain the basis, the vectors, and then by linear combination, by using the linear combination, we can tell which basis vector is how much containing the polymers heat map data. So that this is the reproduce the data by using the NMF data. So this is original data. Then through the NMF treatment, we get the reproduce the data. Okay. So what's the difference from the here raw data and this data? Through this NMF treatments, so this is not quantitative data, but however, the after treatment, NMF treatment, so this z-axis indicated how much containing these polymers, this basis, the vectors. So now that we can tell the how much such kind of the basis vectors containing this original heat map data. Okay, so that this is to prove that this our concept. So that we perform the evaluation of the mass spectroscopies. So this is first we made it a mixture of the polystyrene and the PMMA and the ethyl methacrylate. We just mixed together, okay? And then so the red, the star indicated the true data, and then blue is estimated. So that's indicated the S-mem. So that is so the almost consists. So with a good agreement with that. So in good agreement with that. So that means so our calculations, the machine learning is reasonable, reliable. And the surprising thing is so there are several surprising things to us. So one is in these cases, we don't need the reference data. So that means that only you need is the just mixture of these guys. So then, we can estimate it the 0 point. So that is very important. So you don't need the pure reference samples. And also only the subtle change of the methyl group and the ethyl group can be identified, because that is quite difficult by NMRs, methyl, ethyl. So that is quite difficult, even high resolution of the NMR spectroscopies. So as a result, that is estimated and observed the data. So you can see, so about this, okay, less than 100 ppm orders. So we can tell how much containing these polymers. That means okay, so you get the polypropylenes. Oh no, polystyrene. But so if the ppm 100 ppm order contaminated included in the polystyrene, so you can tell. So that is very high accurate. So that is quite difficult by NMR. So you can tell such kind of the small amount of the contamination, you cannot detect it. So that this is the result of the sequence of the polymers. This is copolymers of the styrene and the butyl acrylated. So that we prepare, we polymerize the polymers, and then we monitors the conversion by using the mass spectroscopy, like taking the samples time by time, and then measure that mass spectroscopies. And then so that this is the pentamer, pentat, change of the conversion, pentat fraction versus the convergence. So they first, that's indicated first at the beginning, so that this SSBSB is dominant. But after proceeding the polymerizations, the most dominant species is SSSB, something like this. So we can tell that by our method, machine learning supported mass spectroscopy analysis. However, so that even though so that we say, so this is our results, so but no one believe it, because no one see before the conversion of the pendant. So we converted this pentant data to the triad. So so the reason why we choose these polymers, so this polymer can tell analyzed by NMRs. So luckily, so by comparing with the NMR data, and also there is the theoretical analysis. So we calculated, we converted the PENDAT data to the triad data. So that you can see that those data very good agree with these data. So that means that this data also reliable. So okay, so this is the cartoon of our method. First, I just put samples and pyrolyzed from the room temperature to 600 degrees Celsius, and then make the data of the fragmented and then monitors the data by other heat map data. Okay, and then so that is of course that is a kind of big data, but this is not enough for the NMF analysis. So by changing the composition ratios, we obtain the similar data by changing the composition ratios. That is necessary. We have to do that. And then so we sorted the same sequence, and then we calculated how much sequence containing the random copolymers, short segment containing C. Okay, so how much in these polymers, for example, this sequence is dominant, then this is not sorted. So this is original random copolymers, and then this is the computer generated the well-defined polymers. So that is the that is equal the meaning. So that is very important. So by using this method, we apply for the semiconductors field, and also the automobile and the plastic recycling. So today I want to show you some example of by using our method. So one example, the simple example, as I told you, so our method used the mass spectroscopy by using the heat map, pyrolyzers. So that means that you don't need the solubilities. You can use the insoluble polymers. So to do that, so we prepared the network polymers. So basically, this polymer doesn't dissolve in any organic solvent, and also in the aqueous solutions. By using the precise polymerizations, we change the distance. We change the distance of the cross-linking point. Okay, so the short length of the cross-linking, the long range cross-linking. So we prepare the well-defined network polymers. Okay, and then we perform that mass spectroscopy and then analyze by using the NMF, and then this is the correlation between the predicted molecular weight of this distance distance between the cross-linking point. So this shows a very good correlation between the predicted data and the known data. So our method can apply for the insoluble polymers. We can tell the molecular weight of insoluble polymers. And also from viewpoint of application, so we use the analysis of the adhesives, because the adhesives are commonly used in anywhere to make infrastructures and the computer or whatever, the automobiles. However, sometimes this causes a very serious incident, problem, and also the accident. Okay, so especially in Japan, so we had a very big tragedy. So in the highways, so they suddenly dropped the seal, and then cracked the cars. So this is a quite serious accident that happened almost 10 years ago. So to analyze the reason why, what is the problem of the adhesives? So the adhesive could not cure the properly. So the properly during the making this tunnels. So something happened. That is problem. That is the reason why this accident happened. So the important thing is they understand the performance of the adhesive after the process, after curing. But they're usually quite difficult. So because once the adhesive cured, you cannot. It's quite difficult to analyze the chemical structures. Okay. So we use our method to analyze the performance of adhesive after curing network polymers. Okay. So as a demonstration, so we use the very common two-part epoxy adhesives with the diamine systems. So by changing the composition ratios, the process conditions, so we get the several different type of the adhesives with different adhesive strengths. Okay. So by using these data, we got the dot mass spectroscopy data. So that's shown here. So that there is some difference you can see. So that means of course, it's quite difficult by characterize these structures, network structures. But by taking mass spectroscopy, so somehow you can tell the difference of the adhesive chemical structures. And then by using our NMF analysis, so we get this kind of the basis vectors, and then we perform the multivariate analysis by using this. So as shown here, so by using these mass spectroscopies, so we can tell which mass spectroscopy shows the good adhesive properties or the bad good adhesive property or bad adhesive properties. So we can tell after curing, after networking of the polymers. Also, another example is here as an application of the polymer sequencer is the in composite materials. So as I told you before, our method can apply for the inorganic materials. Okay. Composite materials with inorganic materials. So as a demonstration, so we performed our method to understand the heat resistance polymers, the performance of the heat resistance polymers. So that usually this heat resistance polymers require the good heat resistance as well as adhesive to attach to this substrate. Okay. So however, so that almost 90% weight percent of these materials is aluminums. Okay. Almost no polymers. No polymers. Almost 90% is aluminums. Only the 10 weight percent is just silicons. So how do we tell? So but however, to express the good adhesiveness, so this silicon network is very important. So how do we evaluate these polymers? So we get to successfully classify the heat conductivities and the adhesiveness. So which mass spectral type shows the good high heat conductivities and also the adhesiveness, so that we can tell the performance, multivalent performance of the inorganic composite materials. Let me a little bit change the topics. Okay, 5 minutes. Okay. Okay, so as I told you, so that we are now conducting, we conducted the materials open platforms initiatives from the 2017 to 2023. So that this is the engage in the parallel, the horizontal collaboration with the chemical companies big major company in Japan and the National Institute of Materials, NIMS. So the aim is to the common research theme is the gathering basic data of polymers. Okay, gathering data and also the structuring the database and estimating the physical properties of materials properties by using the machine learning. So the as a target molecules, the polymers, so we focus on the just polypropylene, okay, as shown before. So that we focus on polypropylene, and then so that we perform the we have done the experiment, the many experiments like mechanical properties, thermal properties, chemical properties, whatever. So at the end we get the more than 40,000 data set. So I think that is the largest database of the polypropylene, commercially available polypropylene database. And also the and then after that, so by using these raw data, precious data, because we perform every experiment by using the same experiment apparatus, instrument, and the same person, same method, everything same. So that the very pure data, so we got it. The many data, for more than 40,000 data. And by using this data set, so just I show you the one example. So we get the XRD data and then can successfully predict the mechanical properties, like a tensile test or the Sharpie test of these polymers. Okay, so but unfortunately, this program just quit at the year 2023. However, after that, so the new project, of the cabinet, Japan Cabinet Office just initiated. So just focus on the circular economies, recycling polymers. So in this program, so that we use we again obtaining performing the data curations. And then now that we have the more than 6,000 data already of the recycled data. So collected by the recyclers and also the many, for example, the East Asian countries, whatever, so foreign countries, because we get many data, and also the recyclers. And also then, but in the program of the MOP, so we just take every data by hand. But now we are just changing by using this to use the autonomous experiment. So this is the mechanical properties. So first we prepared this kind of the card size, credit card size, the specimens with 5 specimens, and then put this sample. At first, make the patterns by using the printers, and then put this sample, transfer the sample to the here. So here we just pull the samples. Then also we monitor this species. Then we can evaluate by using the FEM method. So now we are correcting those data by using the autonomous dataset experiment. Okay, like this. Okay, so now, but through this program, so that the kind of the situation change, especially in the Europe. So they regulated the new plastic for the automobile. Yes, car, auto vehicles. So according to this regulations, so if you want to make a new cars, you have to use the 20% of the recycled plastics. Okay, so anymore, so that you cannot use the virgin plastic for the new cars. You have to use the recycled plastic. So that is quite serious problem. But if you look at what kind of polymers used in the car or vehicles, the most common polymer is polypropylene. So we got lucky. So that we already have the fundamental data set, more than 40,000 data set, 42,000 database. Okay, so that is the very important. So that this is a kind of reference papers. So they say to understand the recycled plastics, the most important thing is to understand the virgin, commercially available plastics. Okay, so if you see the catalogs of the companies, of course they sell the molecular weight is like this, but so some cases so that it has the bimodal natures and also the molecular distributions and additives is different. So the importance is a commercially available polymers database. So that is very important to understand the recyclable. So the 0 point is very important to understand the recycled plastic performance. So that we mix together the virgin plastic and the recycled plastics, and then perform their mechanical properties, thermal properties, or whatever, just like the MOP project. And then we, as a demonstration, so we get the XRD data like this of the recycling polymers. And then we got the mechanical properties correlation between the XRD and the mechanical properties, like a tensile test. So as shown here, so this shows the good correlations. That means that we can understand the lifetime of the recycled polymers, okay, by using this method. So that by using our database. So this is, so yes, by using this data set. So I think we can contribute to the these regulations, the circular economy systems. Okay, so this is a conclusion. Thank you very much for your kind attention.
Thank you. I think the common workflow of material design right now is that you have certain application in mind, and then you specify the target properties, and then you try to optimize a material for that. But along the way, you would definitely get many negative results, which probably do have some unique properties, but you don't know how those can be used. And I was wondering if you've tried building like a system or platform that autonomously looks for an application or like many possible applications of a material rather than like, so it's like an inverse workflow of what you would
What
usually
kind
do.
of property, like mechanical properties or thermal properties or different properties, lifetimes?
Everything.
Everything.
Yeah.
Yes. Okay. Everything. Yes, true. But so now we are working to develop the autonomous systems for the one is the tensile test as shown here. So that is also take a long time to measure it. So we try to make some kind of the autonomous apparatus to replace the samples and then measure the automatically change the data. And also the what else? And also lifetimes is also the key target. So that we are performing the accelerated the simulations of the by irradiating the intentionally irradiated the UV light, the waters, and heating, something like this. So we do perform the cycle experiment to understand the what is the lifetime of the materials. And also the important thing is the data, not only the just data set. Now we are thinking to gather the data into our database, so-called RD, Research Data Express. So put in those every data into the systems. So everyone can access to the data set, like with different properties. And then also they perform the machine learning to predict any kind of properties. So that now we are doing such project.
Okay. but I think that's still like doing the point of that would be to get the properties that you would want. But my question is, like, if you have a random property that you don't know what it can be used for, and like maybe like it would be helpful to have a system that where it can autonomously goes to the internet, for example, using LLM.
President of NIMS. We should introduce such kind of systems.
Yeah.
So the autonomously measure the, for example, the chemical structures, the whatevers, blah, 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 blah. Okay.
Okay.
Then, so get the fundamental models. Okay. But our president NIMS cannot understand that.
I see. Thank you.
Yeah, how do you measure the lifetime? Like what sort of modeling?
Okay, so at the right time. So we prepare the different compositions, virgin and combination of the virgin and the recycled polymers. Then we prepare the specimens, and then to perform the sunshine weather meters to accelerate the degradations, and then take samples and then measure the mechanical properties, everything, so step by step. So that is so that we are taking a long time to collect the data.
I think at the beginning you shared a slide where that some data are available and some are not. So this kind of like where you have multiple property, are these data freely available? Like can anybody access, or like this multiple property data set, like to what extent these are available freely? Like anybody can use therefore?
Yes, at this moment, so only the limited members can access to those data. But I think that within the couple, after embargo time, so that we can open the data, to share the data to the membership, to the members. The member is not the member of the project, membership of the RD project, RD data. Okay. So I think that within after this project, and maybe the 3 or 5 years later, you can access to those data.
Thank you.
Do you want to say?
Yeah, sure. Yeah, just may I ask a question? Supercritical fluids. Do you ever interrogate your polymers? You mentioned that like many of them become quickly insoluble and therefore intractable to solution NMR and other spectroscopies too. But I mean, I was just curious if your analytical facilities are equipped to use supercritical fluids to.
Oh, supercritical fluid. Yes, but still some polymer cannot dissolve in the supercritical.
Yeah, true. Yeah.
Depending on the species, like carbon dioxide or whatever. So yeah. Yeah. I think not so. I think that is not perfect. Of course, the supercritical fluid enhance the solubilities. Yes, but still limitation. I think the limitation.
And your comments about sequencing, that's very, first of all, the pyrolysis thing, that's actually really interesting. Yeah, but in terms of like sequential cleavage, like you can do so easily with a protein generating ladders, you would think, right, that if there was a way, if you start with a polymer that's functionalized on one end so you can tether it to a surface, then you could imagine a gradation of effects that would give you sequential cleavage to do the sequencing. I mean, have you ever thought about that?
No, actually no. But some people working on that such kind of project, and so they intentionally introduce some functions into the polymers after modification, after modification, and then they cut it at the specific positions, and they measure by NMR or the mass spectroscopies, MODI, TOF, something like that. So yes, that is one way. And also they put the isotope is another method. Yes, put in the isotope method at the end to read the sequence.
How about polymers with some content inside, like nanocomposites? Can you handle those?
Composite?
Yeah, like epoxy with something inside, like nanoparticles or.
Oh,
like, I
okay.
don't know, just like.
Like this? Or...
Yeah.
Oh, okay.
I understand this one's like also like a composite, but like putting some nanoparticles.
Yes.
So can you handle that?
Oh, anything fine.
Okay.
Yeah. Graphite, nanoparticles, anything fine. So as long as that it doesn't degrade it. So only the polymer degraded or the organic species degraded. Yes.
Naito-san, I'm highly recommended to use LLM. And
Wow.
separated prediction model recently now nonsense, I think. So
You mean NMF?
Yeah, everything unified to the LLM. I highly recommend.
Yes, that's very interesting. So the first we measure the bunch of data of the different type of polymers, and then just putting the whole data into the LLMs, and then
multi property.
right. Yeah, that's quite interesting. So because we already have many data.
Yeah, but please collaborate.
Yeah, you.
But you know this LLM, so polyinfo already exists.
Yeah, I just got the information.
I'm also
Already exists. China.
Yes, just the
By
information.
the way, okay, then. Okay, then. Thank you very much.
Thank you very much.
Okay. Thank you. Yeah. Thank you again.